This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 263, recorded on April 7th, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How's it going down there? You got hot weather?
We have had a bit of weather. I mean, we've had tornado warnings and actual tornadoes the last 2 nights in a row.
Wow.
And it's been pretty windy. And that all those straight line thunderstorms have come through and have caused the appropriate amount of destruction that everyone fears at this time of year.
It's terrible. Yeah. I saw a video of some people who owned a farm, and this big tornado came and just destroyed the whole thing. They went into the basement and they came out and all the houses were gone.
Yeah, that's principally what's been going on here, and everyone reports it sounds like a freight train coming down the road.
Not a sound you want to hear.
No. Well, you live in the Midwest, Petra, so you're even more prone to these scary things.
We've been lucky this spring, but yeah. The first time I heard those sirens when I moved from the East Coast, I was so confused because it sounded like a World War II movie, but now we just go to the basement.
Yeah. That voice you hear is coming from St. Louis, and it's Petra Levin. Welcome back, Petra.
It's good to be here.
Petra, you know, we had, many years ago, we had an episode where we came up with a new kind of tagline. You know, at the beginning, we see the podcast that explores unseen life on Earth using unpronounceable words. You remember that, Michael?
Oh, yes.
I like that because you guys have talked about, I think with Marco Martin, that you guys talked about a bacteria that you can see. So mostly unseen life on Earth, I think, needs to be the new one. Yeah.
You know, there are some viruses now that you can see in a light microscope. They're like over a micron in length, but, you know, the vast majority you need an electron microscope. But the words we use are long and often unpronounceable.
Yes, that is always true.
Owing to us not speaking Latin as our native tongue.
Yeah, indeed. Also joining us today from Small Things Considered, Elio Schechter.
Hey, my pleasure to be here. Thank you.
All right, today we have a snippet in the paper for you. And to start us off, Michael will tell us about lavender oil.
You know, this is a very popular topic amongst the general public. And so when I stumbled across this paper in Letters in Applied Microbiology, and it's from a group out of China who are at the Guangdong Provincial Key Laboratory of Food Quality and Safety in the College of Food Science at the South China Agricultural University in Guangzhou, China, in the Guangdong Laboratory of Lingan Modern Agriculture in the South China Agriculture University, I thought this would be something that would interest our listeners. And the authors are Han, Chen, Zhang, Peng, Zhang, and Zong. And the title of the paper, in its full public access, is The Elimination Effects of Lavender Essential Oil on Listeria Monocytogenes Biofilms Developed at Different Temperatures and the Induction of a VB&C State. Well, we know Listeria monocytogenes is a typical foodborne pathogen that causes next to impossible, hard-to-treat, often fulminant bacterial infections owing to its ability to grow as a facultative intracellular parasite, with its acquisition resulting mainly from its ability to form biofilms and then enter into this VB&C state. And many investigators have confirmed that chemical stressors such as pH, ethanol, chlorine, any typical household cleaners, even antibiotics and other biocides can actually induce this VB&C state. So while you may be thinking you're cleaning surfaces to protect you and your family, that doesn't necessarily happen. The microbe can't be cultivated, and the net consequence is that you ingest it and then you get sick. Now, I apologize up front since this is only a snippet. I don't have time to explore how this remarkable microbe accomplishes its feat of molecular pathogenic gymnastics. But what I have done for you is I've dropped into the show notes an approximate 7-minute video describing this remarkable microbe from Pascal Coussart's laboratory from the Institut Pasteur. And it's also freely available on YouTube. It's a really cool video showing you how Listeria Monocytogenes actually causes illness. And I also like to offer that in the past here on TWiM, we have extolled the wonders of VB&C microbes, specifically in TWiM 179, that was in 2018. And so I've dropped that into the show notes along with a review from Frontiers in Microbiology on all things VB&C. Now, since you're listening and you don't have time to go rush out and read all those papers, recall that VB&C cells are simply characterized by their loss of culturability on routine bacteriological media. That means you simply can't grow them when you plant them on a plate, which impairs their detection by conventional plate count techniques. Now, this leads to an underestimation of total viable cells in environmental, clinical, or even food product samples, and thus having a VB&C population in a community of microbes does pose a risk to public health. If the microbes are in this VB&C state, you will think the product, whether it be ground beef, chicken, or even those ready-to-eat greens like lettuce and spinach, is safe, when in actual fact, it can still cause you to become sick. So again, we discussed the paper by Highmore and colleagues back in 2018, and that was TWiM number 179. So you can see how it's able to accomplish that. And that was again in Salmonella as well as Listeria. But now on to today's paper. So Listeria is a remarkable pathogen in that many of you may recall that it's a psychrophil, meaning it's capable of growing at low temperatures. Actually, it can grow at 4 degrees C and thus plagues the prepared food industry, specifically those industries that are preparing those ready-to-eat luncheon meats like, that turkey that you had for lunch, the roast beef, or ham. And they are ready-to-eat products that are prepared for the consumer. And if the facilities that are used to slice those meats aren't effectively cleaned, you can effectively become ill. And so, as the authors point out in their paper, much of the research on Listeria biofilms has focused on those biofilms that develop at room temperature, typically 18 to 20 degrees, all the way up to 32 degrees centigrade, which is where they begin to characterize things. However, much of the food processing occurs at refrigerated temperatures between 4 and 10 degrees, and little is known about the survival mechanisms of this pathogen at these temperatures, especially when it comes to biofilms. And so what this paper evaluated was the elimination effects of 4 typical antimicrobial agents on mature biofilms of Listeria and the induction process of VB&C cells in these mature biofilms. And the 4 things that they looked at is the lavender essential oil, good old bleach, sodium hypochlorite, lactic acid. And many of us don't actually think of lactate as a sanitizing agent, but it's used quite often in the food industry. And so it's really pretty remarkable that—
Michael, these would be things you use for surfaces, right?
Surfaces. You'd spritz it on.
Okay.
And of course, and the final thing that they used was good old hydrogen peroxide, the stuff that your mother poured on your skin knee. And so their first question, and why I like this paper so well, is because they teach the reader how to measure things very carefully. And oftentimes methods get the short shrift in papers. And so I'm going to go through. The first question they asked is, does lavender oil, what does it do to the viability of mature biofilms formed at various temperatures? And the way they measured this is by simply looking at the optical density of the refraction of light to see whether or not the agent would effectively disrupt the biofilm. So if it disrupted the biofilm, you'd have more transmission of light, and it would effectively give you a higher transmission or a lower OD. If the light is being absorbed by the biofilm, you'll have the higher OD. But before they did that, they first offered us the minimum inhibitory concentration of the agents used. And remember, they used 4. They used lactic acid, lavender essential oil, sodium hypochlorite, and hydrogen peroxide. And the way they determine their MIC is the way we teach it in the undergraduate laboratory. They did 2-fold serial dilutions. They took 100 microliters of Mueller-Hinton broth and 100 microliters of the antimicrobial agent. They're placed in a well, and then they just serial dilute, taking 100 microliters out of the first well, putting it into the second with 100 microliters of media. And the consequence is, of course, you cut the concentration in half. And then they inoculated each well with 40,000 viable bacteria. That is, those bacteria were able to form a colony on a plate. They determined the MIC for lactic acid. It was 0.625 milligrams per mil. The lavender oil was 1.6 volume to volume. And this is one of the quibbles I have with their paper. They never told us the concentration of lactic acid. They never told us the concentration of lavender essential oil. And they never told us the concentration of bleach, whether or not they were using bleach from, Walmart, or whether or not they were using a higher concentration of bleach. And similarly with hydrogen peroxide, were they using the stuff that we all can buy at the drugstores, namely 3%, or were they using 30%? And I scoured the methods trying to find it, but my guess is they were cut for word count, is what my suspicion is. So the first figure evaluated the removal effects as measured by OD. What they did is they placed 5 mils of broth with different concentration of the antimicrobial agents. They used 1/2 the MIC, 2-fold MIC, and 4-fold on top of these mature biofilms that were grown on cover slips. And then they effectively looked at the OD. And what they saw, and they measured these on biofilms that grew at 32 degrees and at 4 degrees. And what you see is the 2 best agents for disrupting the biofilms were the lavender essential oil and sodium hypochlorite. And the figures are pretty easy to go through because they're just simple bar graphs. And you can literally see how well the agents were removing the biofilm from that surface. And they did this by simply looking for an increase in transmission of light. And in fact, lavender oil was able to remove 63% of the biofilm at 1/2 of its MIC, and 72% of this mature confluent biofilm at its MIC, and it went up at 4x the MIC all the way up to 83%. Now, any of you who have ever worked with biofilms know they can really hold on to substances, even if they're dead, because of the extra polymeric substances that are being manufactured by the film. And so I think being able to displace 83% of the biofilm is pretty remarkable. And the authors also offered that lavender oil is also good at removing warm biofilms of Staph aureus from glass and polystyrene surfaces, as well as knocking out Campylobacter. And the reason we worry about Staph and Campylobacter, anyone who's ever eaten poultry know that Campylobacter and poultry go like, eggs and bacon, so to speak. They're always together. And similarly, Staph aureus, when you're using prepared luncheon meats because of the high salt, that favors the growth of staph. Now, moving to the cold box, the lavender oil was also found best at eliminating cold biofilms. But here, the concentration that was able to displace had to be increased. And so this is where the microbiology gets interesting. What's different about growing a biofilm at 10 degrees versus growing one at 30 degrees? And they offered that biofilms developed at colder temperatures often are able to display stronger resistance against those formed at 32 degrees. They offer us no mechanism of how this is happening, but that's, of course, work for you all out in the audience to get busy on, to ask the question, what proteins are being expressed, what genes are being turned on. And they offer us one tidbit. They say the reason cold stress can induce tolerance to antibacterial agents. Now, the next experiment they did is they addressed the effect of the essential oil on the metabolic activities of the mature communities at different temperatures. Now, while the second figure almost looks like a carbon copy of the first, the method that they use to evaluate the biofilm is completely different. They effectively evaluated the metabolic potential of the microbial community. And they used something called an Xtt assay, which is really a quite straightforward and simply ask a simple question to the microbial population. Can the redox dye we have dumped on top of this community accept electrons from the live biofilm? And the redox dye is 2-methoxy, 4-nitro-5-sulfophenol, 4-tetrazolium carboxanilidolide. That's why they abbreviate XTT, another word you can't pronounce. But it's really cool because the assay is asking, do you have a metabolically intact community? If you will, it's actually assessing, do you have an energized membrane? Or an alternative way to look at this, if the community is still viable but non-culturable, you're still moving electrons into the membrane because you're still doing maintenance energy. And if you can measure this very easily simply by asking the question, does the redox dye actually accept the electrons? And when it does, it changes color. So it's really pretty easy. And again, this figure is really simple and straightforward to evaluate. You simply look at the removal rate, and the higher the bar graph goes, the better the agent is at removing. And again, the two great agents were the lavender oil and bleach. Now, folks don't like bleach in a work environment because it has a tendency to burn your lungs, especially if you're working a 10 to 12 hour shift. And no one likes working in the swimming pool environment that the bleach actually conveys. And again, the lavender oil showed its effects regardless of whether it was the warm biofilm at 32 degrees or whether or not it was the cold biofilm. Now, moreover, they offered that not only was the community metabolically diminished in concert with what they observed for the first optical density figure, they also offered it was consistent with its eradication effect on biomass, indicating that the lavender oil could indeed displace or, if you will, destroy the mature biofilms of the Listeria Monocytogenes because, dead microbes don't donate electrons to redox dyes. In their combination results and discussion, they offer an interesting tidbit for us, suggesting that bleach and peroxide may only be good for disinfecting the tops of a biofilm. And because of the lavender oil's oil potential, it's able to penetrate and get into the inner biofilm, which they speculate is potentially why the lavender oil was better than bleach or peroxide, both hydrophilic compounds, at penetrating the biofilm. And so then in the next experiment, they ask, can the lavender oil penetrate? And here they ask the question by doing yet another assay. And they are doing it by asking what's going on with the nucleic acid of the microbe. And they're using a really clever technique that some of you may know. They're using propydium monoazide, or PMA. Now, if you have an intact membrane, the cell won't take up PMA. If you don't have an intact membrane and you've been killed or damaged, or your membrane has a hole in it, then the PMA will freely diffuse across. And then if you shine a little halogen light on it for about 5 minutes, the PMA will intercalate into the double-strand helix of the microbe's DNA and cross-link. And the consequence is now the cross-linked nucleic acid will not be able to melt in the PCR, and thus you can discriminate between live bacteria and dead bacteria because the DNA of the dead microbes has been effectively inactivated or rendered refractory to the PCR assay. And this was a really cool experiment. And so in this experiment, they used 4x of the MIC of the lavender oil to really get it to penetrate. And they asked how many cells were able to grow and how many had an energized membrane or were considered to be operationally viable. Now, here the figures change a little bit, and you got to put your thinking caps on. A viable cell implies an energized membrane. If you're able to culture the cell, that is effectively means it will grow up on a Petri plate and you can count it with your finger. And when treated with 4x of the MIC of the lavender oil, the number of viable and culturable cells in the warm biofilm decreased by 6 logs and 9 logs respectively. So this was a little bit backwards to me because I thought, the lavender oil would actually kill the Listeria, but it actually just caused them to, I guess, displace because they had more viable cells or cells with an energized membrane, or the PMA wasn't able to get in to the nucleic acid. But nevertheless, they think this is still a pretty remarkable result because the viable cells and the cultured cells are down by so many log units. Bottom lining this, the lavender oil was more effective at reducing the viable cells compared to bleach, and it was found to be dose dependent. Now, again, here's where the microbiology gets a little bit interesting to me. After the lavender oil treatment, the number and ranges of VB and C states in the warm biofilm and cold biofilm, here's where it really begins to crater. And once the cells have entered the VB and C state, they then have this decreased susceptibility to antimicrobial agents, and this then can contribute to relapsing and chronic infections. And since this was a combo results discussion section, they bring in a narrative about Vibrio Vulnificus also displaying this behavior, in that when Vulnificus, a gram-negative microbe that causes food poisoning as well, often associated with shellfish from the Gulf of Mexico, it's actually protected against various potentially lethal challenges such as strong oxidants, a low pH, osmotic perturbations, ethanol, heat, antibiotics, and even heavy metals. Now, finally, no self-respecting study of biofilms would be complete without a few experiments involving confocal laser scanning microscopy. And the presentation of their images, I encourage you to go look at them. They're really quite remarkable. They're really easy to figure out because you've already deciphered how they're measuring things. They're using the magic redox dyes, and here the experiments they conducted confirmed that the treated biofilms became thinner, the structure was more sparse, and their appearance was blurry, which is suggestive that the integrity of the biofilm community, normally very ordered and structured, is decaying. So bottom lining it again, in addition, the high levels of the lavender oil and the sodium hypochlorite or bleach at 2 and 4 times the concentrations of the MIC could significantly reduce the living cells in mature biofilms, and most importantly, from a food sanitation issue, promote the dispersion of biofilms into planktonic cells so that when you hose down the surfaces after your shift, you can actually wash away the offenders such that they don't grow back at night. Remember, you're in a cold box. You're effectively carving these meats in the cold. And even though your shift is over, you want to make certain that the biofilm doesn't come back to contaminate that beautiful boar's head turkey that you're going to eat the next day for lunch. And what finally was fascinating to me was that the biofilms that developed at 10 degrees were less susceptible to these sanitizers than those formed at 32 degrees. So I'm really interested to hope that this group of authors actually investigate what's going on with Listeria. The good news was that lavender oil works. It offered a greater opportunity to facilitate a significant dispersing effect on the biofilm cells. And going back to why does all this matter? Well, our current methods of releasing produce or releasing prepared meats is dependent upon culture rather than more expensive PCR or even the Xtt redox testing. And so what I would really challenge the people who are responsible for our food safety is I'd like to see a full-scale, if you will, trial done in food processing plants to see if in a mixed and active environment where food is actually being prepared, whether or not lavender oil will work or whether or not, God forbid, we develop resistance to lavender oil. And so all of those things come up. Summing up, lavender oil presented the most remarkable elimination effect on the Listeria biofilms among the tested microbial agents. Moreover, the numbers of viable and culturable cells in the mature biofilms decreased notably with the lavender oil treatment. But again, the caveats are we don't know if the population will become resistant. How easy is it to become resistant to lavender oil? So don't go out there and buy lavender oil futures just yet. We have to wait to see what will happen.
Michael, what is it about lavender oil that disrupts the biofilm?
I don't know. I think it's maybe it changes the octanol water coefficient of how the community is attaching. It may have something to do with how the Listeria are attaching to surfaces. So all of those variables.
Presumably there is some component. It's probably a complex mixture, this essential oil. It's not just a single, right? Yeah.
It's just not flower petals.
Yeah.
Yeah, I've always wondered with these essential oils what they are exactly. Are they squeezing it out of like you get out, like olives, or do they infuse mineral oil or something with
lavender
oil?
flowers?
And was there control where they just use the base oil, or is it all?
No, there was no base oil control. And that's where I took them to task. I mean, they didn't tell us the true concentration of the peroxide, the bleach, the lactic acid, because the lactic acid that's on my shelf in the lab is 40%. But I think we had to specifically buy that concentration. I'm a molar guy. I want to know moles per liter.
Yeah.
Because I can do the calculus on moles per liter to know how many molecules are actually present.
100% molar.
Yes.
My other question was the viable but non-culturable. I think it's important for our listeners to understand that those generally do not start growing again. That when we use that term, we mean that they're alive in that they are metabolically active, but they can't reproduce is the general understanding.
But they do. That was TWiM 179. Keevil and Highmore actually showed that the Listeria and Salmonella VB&C cells can actually infect C. elegans and actually cause an infection in the gut of the animals that the VB&C cells are ingested by.
So the idea is that they just need to be under the right circumstances. They're just VB&C in the lab, but if they get into an animal,
and they actually run amok.
That's scary also because cholera has these VB&Cs, which are quite famous,
yes.
But it's still, as far as I know, unclear what.
Well, cholera gets even more squirrely because of the phage.
Yes, exactly. The phage.
Michael, what is this going to be now used in the food industry, you think?
I believe it will be.
Huh.
I believe it will be.
Next time I see lavender, and we have lots of lavender here, I'll think of this. All right. Thank you, Michael. And now we have a paper from Petra.
Okay. So this paper was published just recently in eLife. It's titled Inhibiting Host Protein Deposition on Urinary Catheters Reduces Associated Urinary Tract Infections. It's by Marissa Anderson, Chun-Ki Fong, Alyssa Ann Labella, Jonathan Molina, Alex Molisan, Matt Champion, Caitlin Howell, and Ana Flores Morales. The last two are corresponding. The first author, Ms. Anderson, is a PhD student in the Flores Morales lab at Notre Dame. She writes that while she started university as an art major, she found herself interested in the STEM classes she had to take as part of her college liberal arts curriculum. And I feel that is a total plug for liberal arts. I went to liberal arts college too. And I had to take art. So in another serendipitous event in her life, although she started her PhD wanting to do neurobiology, all the labs that did that at Notre Dame, I guess, were full. She decided to work in Dr. Flores Morales' group, which was just starting at the time. She was really intrigued by the clinical and translational aspects of her program. Ms. Anderson had never taken micro in her life. I totally identify with this because when I started grad school, I thought I was going to do frog development, and I ended up doing microbiology, and I didn't take micro until I taught micro. So I completely 100% understand this path. She really wanted to make sure to thank not only other members of the Flores Morales lab, but also their collaborators in the Howell lab in the Department of Chemical and Biological Engineering at the University of Maine. Among other interests, the Howell lab has expertise in treatments to prevent biofouling, biofilm formation on surfaces, and that's really the focus of the paper that I'm going to discuss. When I asked Dr. Anderson about a strong memory of an especially exciting day in lab, she said there are lots of exciting days in the Flores Morales lab, but the day they figured out that liquid-infused silicone, which is the sort of focus of this paper, was by far the best of all the modifications they've been testing to prevent bacterial growth on catheters and binding to fibrinogen, was one of her favorite, as the results were so strong they blew away all their previous work, which is always great when you see a very strong result. Acceptance of this paper was also a great day. They'd been editorially rejected from other high-impact journals. I think many of us can identify with that. So getting the notification from eLife, it was being sent out for review, was awesome. Ms. Anderson says most days are pretty interesting, and I think that's a great outlook. There's always something. Once it was a mouse with an obstructed ureter and its kidney was the size of the head of the mouse. But she also finds it exciting when someone gets a protocol to work. And again, I totally identify with that. When asked for advice, she would give herself as a younger scientist. She said she would take more lab courses, take a gap year, apply for jobs that interest you, even if you aren't 100% qualified. The worst that can happen is you get a no, which I think is something we can all be a little more brave if the worst that can happen, or just remind ourselves that no is not the end. It's actually a beginning of a new project. And my favorite piece of her advice is something that I think all of us can keep in mind, which is to remember to take care of yourself so you can do your job well. So self-care. And finally, she has some outside interests, but the one that she highlighted is spending the day outside hiking with her 1-year-old golden retriever, Riley, which is also a great way to spend a day.
Cool.
Yeah. So it was exciting. She wrote a lot, and I really appreciate all this insight. So the paper, Michael was just talking about biofilms. And as a reminder, biofilms probably I think I read the 70% of bacteria are in biofilms in the environment of some kind. So they like to settle on surfaces, pretty much any kind of surface. And they make a matrix. Often it's a polysaccharide that helps the bacteria stick together and also helps them kind of stick to the surface. And these biofilms develop multi-layer, multicellular structures. Some of them can be quite elaborate. They're very difficult to remove, as Michael noted. And because many of the cells in biofilms are in low metabolic states or quiescent, they're resistant to antibiotics. It's also important to note that in the body, when biofilms form, the immune system has a very difficult time dealing with them because there are multiple layers and the antibodies can't really get in deeper. Biofilms are a problem not only for human health, they're a problem for lots of things. I always remember the Navy spends a lot of money on engineering the hulls of ships to prevent biofilm formation because they're a huge issue. And they usually start with bacteria and algae, which provide sort of a surface for other larger organisms, and finally barnacles and other crustaceans to stick to. You can imagine if you have a smooth hull and then you get all these barnacles and bumpy things on it, that causes a huge drag, which can also damage the hulls, but it reduces fuel efficiency. And when you're got these big aircraft carriers and other ships, it's big bucks. And a lot of work in the sort of industrial side of biofilms is finding ways to reduce biofouling, which can also occur in, for example, pipes. Water pipes are a big problem with biofouling. But the focus of this paper is really in humans. And biofilms are really a problem in hospitals for indwelling medical devices. They can be, people who are no longer in the hospital, like heart valves. But the really big issue is catheters. So if you're in the ICU, people become catheterized, which makes it easier on one hand. But the problem with these catheters is they increase inflammation in the bladder. They're often left in for a really long time. And previous work from actually Dr. Flores Morales, when she was actually a postdoc, she found that typically catheters induce inflammation in the bladder, which leads to a production of a protein called fibrinogen, which is also called host clotting factor 1. And the fibrinogen actually comes from the bladder and coats the inside of the catheter, and then bacteria stick to the fibrinogen. So they're not sticking directly to the catheter. They're sticking to the fibrinogen, the protein on the surface, in her earlier work. And she actually, in this early work, showed that Enterococcus faecalis and Staph aureus actually have special adhesin proteins on their surface that bind for fibrinogen. So it gives them a hold, not only in the catheter, but actually fibrinogen. Any wound actually is going to have some of this stuff. And in the bladder, an inflamed bladder, like maybe a bladder infection, it's also going to have some fibrinogen, and these bugs can stick to that. And once they stick, you get the biofilm development. You can get it in the catheter and into the bladder, get bladder infections, which are, if you've had one, not pleasant. But the real danger is that they can proceed up to the kidney, and kidney infections often become bloodstream infections, and they're really hard to treat. Again, they're not a lot of quiescent cells, so that makes them resistant to the immune system and antibiotics. But once they're in the bloodstream, it's even more difficult. And she had these incredible statistics. 40% of all hospital-acquired infections are from catheter-associated UTIs, CAUTI, CAUTI. And once the pathogens are in the bloodstream, there's a 30% mortality rate. So this is a huge, huge problem, especially again for patients who are catheterized in the ICU. They can't move. They often can't communicate that they even have the bladder infection in the first place. The standard practice is removing the catheter once there's a UTI, replacing it, and giving antibiotics. But this is often too late because there already are biofilms in the bladder, and it's very hard to treat. So the Flores Morales groups, one of their main things they want to do is try and figure out how to treat or prevent these infections. And the Howell lab, again, works to find strategies from an engineering perspective that also inhibit formation of biofilms. So it was a great pairing of these two groups. And this focus of this is really to inhibit fibrinogen deposition and then biofilm formation. So again, the bacteria really stick. They seem to need to stick well the fibrinogen first. And so they wanted to go after the fibrinogen. So they started, as I alluded to in my description, in Ms. Anderson's description, just by looking at different catheter materials coated with different things. And as I noted, as Ms. Anderson was so excited by, liquid-infused silicone, LIS, it's basically was the best. So it's basically a silicone catheter, so a sort of standard kind of catheter. And it's been soaked in silicone oil, medical grade. So this is used sometimes to reduce friction in certain situations where you might have a medical device or something that might be present for a long time and cause irritation on the skin. Anyways, if they soak the catheter in that until saturated, and they have these nice saturation curves where they're measuring the weight to see when it's saturated, they showed that it works to inhibit both fibrinogen binding and binding of a range of urinary tract pathogens. So there's previous work, actually, from the Howell group and others, showed that liquid-infused silicone, basically soaking the catheters or materials in this silicone oil, actually inhibits bacterial growth. And this paper is actually trying to take it one step further to see if this can actually work in an animal. And I love this paper. It was really logical. It's beautiful. It's easy to follow. So they first start with identifying neuropathogens that interact with fibrinogen during catheter-associated urinary tract infections. So there was a long list of pathogens that's known to cause infections in people who have catheters, but it's never been systematically studied, especially in the mouse. So they tested not only Enterococcus faecalis and E. coli and staph, which they've done before, but also Pseudomonas aeruginosa, Klebsiella, Nemophila, Acinetobacter baumannii, and they used one yeast, Candida albicans. They used a mouse catheter model. This was actually Dr. Flores Morales helped develop this. It's basically you put a tiny piece of catheter directly into the bladder of a mouse. You can't really catheterize a mouse very easily, so you just put this little catheter piece into the bladder, and then you add bacteria and find, it's really quite straightforward. And they find that all the pathogens that they use in this model, within 24 hours, they have the pathogens stick to the catheter, and they find that binding is coincident with fibrinogen binding. And this is in figure 1A and 1B. And I think they do fluorescence microscopy to really show, or imaging to show that the fibrinogen stain and the bacterial stain match up. They next, after establishing their sort of model system, they move in vitro to show that fibrinogen binding enhances binding of the same set of pathogens to the urinary catheter material. So again, the model is that fibrinogen binds first, and that gives sort of a surface for the bacteria to stick. And this is in their figure 2, which is also beautiful. They coat the catheter with fibrinogen or uncoated catheter material, or they coat it with bovine serum albumin sort of as their control protein. And then they look at the binding of the pathogen. So they have uncoated, fibrinogen coated, and BSA coated catheter material. They set the binding of the fibrinogen to 100%. So the pathogen binding to fibrinogen to 100%, and they look at relative binding to uncoated and BSA coated. And they find it's actually really clear that the fibrinogen really promotes pathogen binding. The BSA promoted it to some extent relative to uncoated, but the fibrinogen is usually about twice as high, better at getting pathogens to bind, or worse, depending on your viewpoint. This figure actually has the two panels that shows how long it takes to infuse the different catheter materials. It seems to reach maximal in under an hour for tubes and under 10 minutes for mouse catheters. So basically, they're just submerging them in the silicone oil. It's super simple. Next, they showed that the liquid-infused silicone treatment, submerging the catheters in the silicone oil, not only prevents fibrinogen binding in vitro, so it inhibits the binding of the protein, it also prevents pathogen adhesion. So this makes sense, right? Without the fibrinogen, you're not going to get the pathogen binding, and at least not efficiently. And for this experiment, this is in vitro. They grow cells in sterile urine supplemented with BSA in the presence of the catheter material. Okay, this is all great. This is all set up. So the real experiment is really to show that the liquid-infused silicone prevents pathogen binding and ideally fibrinogen binding inside in their model system. So they catheterize mice with unmodified catheters and these liquid-infused catheters. They add the pathogen, and then they remove the catheters and they examine them. And they find that colonization on the liquid-infused silicone catheters was significantly reduced regardless of the infecting pathogens. And if you look at figure 4 and you look at the fluorescence that shows you the fibrinogen on the catheter and the bacteria on the catheters after they've been removed from the mice, the LIS catheters, these liquid-infused silicone catheters, are almost completely blank. They're almost completely black. It's very impressive.
How many days after implantation?
I think it's 24 hours. Yeah, 24 hours.
24 hours, okay.
Yeah, it's very...
because they colonize so fast, you don't need to go out, the 40 days that the previous paper was looking at.
Right, you don't need to go out. They're putting a good number, I think it's 10 to the 7th bacteria into the... Yeah. It's really to look at how quickly they form on the catheter. So for Pseudomonas, Acinetobacter, and E. coli, they see lower numbers in the kidneys. That's a really good sign. For E. coli and Candida, they see lower numbers in the spleen, really low numbers in the spleen of the mice where they use the liquid infused catheters versus the untreated. And the spleen is really important because this is evidence that there's a lower probability of a bloodstream infection. So that is great. And again, they have data for each mouse, but it's really easy to look at and see what's going on in the fluorescence images of the catheters where you can see the fibrinogen and the pathogens in figure 4. It's really cool, beautiful data. The next thing they go on to, and I find this really fascinating, and I feel like there's a lot more work to be done, is, so remember at the beginning I mentioned that the fibrinogen is part of the inflammatory response, that the catheters cause inflammation, and that leads to production of the fibrinogen by the host. And in figure 5, they look at inflammation in particular. So they put in the liquid infused catheters and the untreated controls and the bacteria, and A through G is histology sections, which I'm not used to looking at those, but people who are, I think it should be pretty clear there's less inflammation, but H through M are fluorescence staining, and they stain for both the pathogen in red. Green is fibrinogen, which remember is sort of an indirect marker for inflammation. And they're also staining for the neutrophils, which are also coincident with the infection, and that's white. And this is really clear if you look at the E. coli UTI 89, which is a uropathogenic E. coli, panel I, and the Acinetobacter panel L, and the Candida panel M. That mice that have been infected but have the liquid infused silicone catheters are essentially they almost look as if they have no bacteria and no catheter. And
it's night and day.
It's really
It's
clear.
Night and day. You don't have to be a histologist to figure out panel L.
Yes.
You look at the left panel, it looks like your thumb is literally in the middle of the figure. And then you look on the right-hand panel, and it's wide open, flowing. So the kidney is actually going to do what it needs to do.
Yeah.
Move liquid.
It's very impressive. And it's very impressive. And the reduction in inflammation, again, is really cool. And I guess I wasn't expecting that, because it's the inflammation that leads to the production of fibrinogen that provides the surface on the catheters for the pathogens. And they're finding not that the fibrinogen is or isn't binding to the catheter, but that it's probably not being produced in the first place because the liquid infused silicone, the silicone oil that's infused into the catheters, is somehow preventing the inflammatory response, which is, I think, a surprising result. I was surprised by that, because you're still sticking a piece of silicone into a tiny bladder, but for some reason the liquid infusion, maybe it's softer, is unclear, but it inhibits the inflammatory response. And it suggests,
which is good.
Oh, it's
awesome.
Is
bad.
Yeah, inflammation is bad. Yeah, exactly. And that suggests that where there's deposition of, it might be that normally in a normal untreated catheter, where there's deposition of the inflammation leads to fibrinogen deposition, which leads to biofilm, which enhances the inflammatory response, that there might be some kind of vicious cycle. And so by breaking the inflammatory response, you may be solving all of these problems at once. It's really exciting.
And looking at that one figure on, which is page 44 of their, I guess it's the pre-production version of their paper, it shows you, the reader, how they assembled the data and how things are then merged on top of things. And you can see where the neutrophils, and which is effectively where the fibrinogen is going to be deposited on, because the neutrophils are going to the source, and that's effectively where the inflammatory response, or the fibrinogen, is going to be laid down. And when you see the merge event, it really makes it crystal clear to the reader.
Yeah, it's so pretty.
It is. It's so pretty. I think that's the perfect sentence.
Yeah, it's so pretty. And again, I think this is all really exciting. Anytime you can prevent a biofilm and antibacterial resistant, either intrinsically or otherwise, bacteria from growing is great, but that it's reducing the inflammation is incredible. And then they do some proteomics to show there's generally less host protein deposition on the liquid infused silicone catheters. And that, I think, was really nice, because in previous work they'd shown it was fibrinogen, but this says there are also other proteins that could provide the foothold for the bacteria. My favorite image is actually figure 7. And so one thing about eLife, which I enjoy, is that you can read the reviews. So anybody looking up this paper can see the reviews. And in reading the sort of summary of all the reviews from the editor, one of the requests was that they include a sort of graphical abstract. Sometimes these are at the beginning, they put theirs at the end. And it shows the difference between what's happening in really nice cartoons between the liquid infused catheters and the untreated or catheters. And in terms of persistence and dissemination, it's really beautiful. If you read anything about this figure, you can pick up paper, you can just look at figure 7. And I just thought it was super helpful. So I just want to speculate a little bit. One is, and I think this is actually very doable, they don't know what most of the pathogens are binding to, other than the work that had been done by Dr. Flores Morales previously to identify the adhesins for staph and for enterococcus. But in the case of E. coli and Klebsiella, there's some evidence that it might be type 1 pili, which are known to bind to mannosilated receptors in the bladder epithelium. So fibrinogen is glycosylated, so they speculate that maybe type 1 pili from these two related bacteria might be binding that way to the glycosylated residues on fibrinogen. It's also exciting because previous work has shown, people have tried finding things that prevent biofilm formation and binding to catheter material in previous work, but they'd always been able to show it works in vitro, but not in vivo. So, for example, you could embed antimicrobial compounds or bacteriostatic compounds into the catheter material, and this would prevent growth in vitro. But when you tried to move into a mouse or another animal model, it didn't make any difference. And the authors speculate maybe these treatments are less robust, don't work well in the complex in vivo environment. It could be that there's breakdown of these things that you're infusing into the catheters. So in this case, the silicone oil, at least over 24 hours, seems to be quite robust. And so that's great. I think the big take-home message, one of the big take-home messages is that you got to prevent the protein deposition. That's kind of the missing link. Don't worry about the bacteria, worry about what they're binding to. And so they suggest actually that protein deposition is the Achilles heel in this situation. I think, again, this idea of preventing inflammation is super important. I mean, obviously, once you have an infection, you need to get the inflammatory response. But if the inflammatory response is actually promoting the infection, you want to avoid the inflammatory response. And I think that's really exciting. And it looks like they're going to have a lot of work to do, which is always exciting if you're especially a newer PI, but I get excited too.
So they say that to translate this to humans, we need to understand the effects of silicone oil on the immune response.
Absolutely.
And understand the impact of swelling on manufacturing, because when you soak it, I guess the catheters swell, right?
Right. And you certainly want pre-swelled catheters before you insert them, I would guess.
So they would be manufactured and packaged with the silicone oil, I presume, because at bedside, you don't want to be infusing it.
No. All sorts of sterility issues. That would be my guess, that you would come packaged in the silicone oil, for sure.
What is this idea about the impact of silicone on the immune response? What are they worried about there?
It could be a stimulant.
Further inflammation, yeah.
Remember, what do you put in adjuvants of vaccines? I mean,
you stimulate inflammation.
Quailing.
Yeah, that's right. So even though bacterial colonization is impacted, there's no biofilm, you're still worried that the oil is going to somehow cause damaging inflammation, right?
And it's the inflammation that leads to the deposition of the fibrinogen. So by virtue that you're putting the foreign object in with the silicone oil, even absent bacteria, if the fibrinogen gets laid down, then the bacteria that are just normally passing through the system could effectively find a nidus in order to attach themselves. Mm-hmm. I mean, this is the big problem with infections. And I think in both papers today, we see that microbes are really good at forming biofilms. You just have to give them an opportunity and they'll do it. And, this is as an alternative to using antimicrobial therapy. And the question that I had for both papers is silicone oil inhibiting the inflammation similar to what the lavender oil is doing by disrupting the biofilm? And would the silicone oil also disrupt the biofilm in an already established infection? Effectively happens? Because normally when someone is catheterized and they develop a UTI, the standard of care is you remove the offending urinary catheter and you put a brand new one in. But the infection is still in there and it can seed the new catheter coming in. And so if the silicone oil can then diffuse away and begin to displace the biofilm that's on the bladder wall, that would be really cool.
Right. No, that's a really good question. Do we know this paper didn't ask this question. So
no,
but does silicone oil soothe inflammation? Because if it soothes inflammation, then that's why that the bladder does react to having a catheter in it, which I would think it would no matter what. But the silicone oil acts as an anti-inflammatory. But it also raises a question, could you use a different anti-inflammatory other than silicone? I mean, because it is the bladder, so in a way, it's easier to manipulate than other parts of the body.
Oh, yes.
So that's a really, maybe there's even a beverage that's anti-inflammatory. I don't want to go down that road, but
no, don't go down that road.
But that's a really interesting question. So,
well, that's cool. Very good story. Thank you, Petra. All right, let me read one email before we wrap this up. This is from Nick, who writes, I was surprised at the end of a recent episode that you said you don't get emails. I don't have anything related to microbiology to share with you, but I wanted to drop a note of thanks. In particular, I thank you to Elio. Your book, In the Company of Mushrooms, started my conversion from a business IT person to a hopeful microbiologist. I've gone back to school. I'm almost done with my prereqs to apply for a graduate program in mycology. It's been a blast. I just finished an internship looking at ACE2 independent entry methods for SARS-CoV-2, but that's more TWiV. Regardless, it has sparked my interest even more. Part of my interest may be inherent in my genes, as my dad was a certified master pastry chef and did intensive study with French bread makers, cultivating an inherent love and interest in mycology. Not that he knew the word, He always said that cooking is an art and baking is a science. In an effort to not attribute my interests to one parent over another, I feel I should also give credit to Mom for encouraging me to think small, literally. She has been pursuing a new hobby in her later years of macro photography. The amount of interesting things that she is able to find in just her backyard or even just in a square foot continues to astound me. Small things considered, indeed. Here's a link for the curious to her Instagram.
Thank you so much. I appreciate that. Very kind.
It's good to hear when you do good things, right, Elio?
Why not?
You do many good things, Elio.
Overall, thank you all again for the continued education and entertainment. Sincerely, Nick, Sunny, and 77F here in Dallas-Fort Worth, coming just after a week of rainy coldness. This is the golden time of the year after winter cold and before summer heat. All right, and then I'm going to read one more short one. We have a longer one, which I will save to next time. This is from Matt, who writes, Hi there. I was listening to the carnivorous vulture bees TWiM and heard Elio mention that someone should look at the Pear Microbiome. I thought you folks might be interested in a paper my colleagues and I published in 2021, Orchard Management and Landscape Context Mediate the Pear Floral Microbiome.
Wow, I missed that.
And applied in environmental microbiology. So he provides a link. This is from Matt, one of the,
Thank you, Matt.
The authors on this paper. So there you go, Elio. You ask and you get. Our listeners deliver.
Ask and you shall receive.
Yeah, not you say jump and I say how high. All right. That'll do it for TWiM 263. The show notes are at microbe.tv/TWiM. You can send your questions, comments, twim@microbe.tv. We do enjoy hearing from you. Even though we know lots of people are listening, somehow when you write, it really emphasizes that. twim@microbe.tv. And if you enjoy what we do, please consider supporting us financially. You can go to microbe.tv/contribute. We are a 501c3, Microbe TV, that is, and your contributions are U.S. federal tax deductible. Elio Schechter is at Small Things Considered. Thank you, Elio.
My pleasure. Thank you.
Michael Schmidt at the Medical University of South Carolina. Thanks, Michael.
Thanks, everyone.
Petra Levins at Washington University in St. Louis. Thanks, Petra.
Thank you.
And I am Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.